Thank you. My name is Peng from ASI. Today I will talk about immunogenicity assessment during the development of antibody therapeutics and how we work to ensure safety and both safety and efficacy. Today I will talk about immunogenicity because it's one of key challenges that differentiate biologicals from small molecule drugs. Even highly optimized antibodies can trigger immune response that impact efficacy, safety, or long-term usability. So here I will share how we actively assess management immunogenicity risk during development with a practical example from our recent project and collaboration with our partner ProImmune. This is topics for today's presentation. I will start with the background, then I will introduce the project overview and our current development stage. Next, I will explain our integrated immunogenicity risk assessment strategy, followed by details of our project ASI immune collaboration using in vitro immune assays. Finally, I will summarize the key takeaways and discuss the business value from a cost-benefit perspective, and conclude with a Q&A session. Thank you. So here is the background. As you all know, immunogenicity assessment is an essential part of antibody drug development. Immune responses against therapeutic antibodies can significantly affect both safety and efficacy. Anti-drug antibodies may reduce drug exposure, neutralize pharmacological activity, or cause adverse immune reactions in patients. Importantly, studies show that the majority of therapeutic proteins may induce some degree of immunogenicity. This means immunogenicity is not an exception, it is a rule. For this reason, and our ASI, immunogenicity risk evaluation is embedded early into discovery and lead optimization workflows. In today's presentation, I will focus on practical methodologies and lessons learned from proactively manage immunogenicity risk rather than reacting to problems late in our development. This slide provides just a context from current industry perspectives on immunogenicity risk assessment. Across the industry, this data shows that there is a clear shift toward earlier and more systemic immunogenicity evaluation. Rather than treating immunogenicity as late-stage clinical issue, many companies are increasingly integrating in silico prediction and in vitro immune assays and developability assessments and the discovery very early stage. This approach allows earlier risk identification and more informed candidate selection, reducing the likelihood of costly downstream failures. This slide summarized our project overview and development progress. Immunogenicity risk assessment was integrated into drug discovery progress, precise through the multiple layers. We applied in silico T cell epitope scoring, then followed by the in vitro DC T cell assays, and together with pharmacodynamic and biophysical characterization. Through collaboration with ProImmune and other partners, we successfully selected two lead candidates. Rather than treating immunogenicity as a downstream concern, it became an complexity criterion for the lead selections. So finally, as a result, our project has now processed into the development stage with increased confidence and in the selected two antibody molecules. This slide that illustrates our integrated immunogenicity risk assessment strategy. Firstly, we use in silico epitope prediction allows rapid sequencing of antibody sequencing for potential T cell epitopes from across HLA class 2 alleles. This helps prioritize the candidates early, many candidates, 200, 300 candidates early. Second, in vitro DCT cell assay, we use this assay. This assay provide biological relevance by experimentally evaluating antigen-specific CD4 positive T cell response by using human donor samples. And third, immunogenicity data integrated with PK, PD, pharmacodynamic, and biophysical characterization. So no single assay is sufficient on its own, but together they provide a holistic and decision-ready immunogenicity risk assessment. So this integrated approach supports confident decision-making and significantly reduce the risk of a late-stage failure due to confidentiality. Here, the specific data are not shown here, just a positive, as you see in the first and second lane, assay control 1, assay control 2 is PPD and KOH. It can induce 100% immunogenicity. Next slide. In this slide, just highlights our collaboration with ProImmune. ProImmune company provided a specialized external expertise, including this assays to a diverse HLA typed panel representing HLA DR, DP, and DQ in the last figure, you can see from in the DCT cell assay, we first use a CFSE labeled PBMC. This PBMC will co-controlled with antigen-loaded dendritic cells, and then the CD4 positive T cells proliferation was measured by using the flow cytometry. From operational perspective, communication was very efficient, and the data was delivered within approximately 8 weeks. This timely execution was critical, not only sufficiently, but also programmatically and directly contributes to our project to achieving an important discovery milestone. To summarize the key takeaways, so early and systemic immunogenicity risk assessment is critical, enable of successful antibody therapeutic development, integrating in silico prediction, in vitro immune assays, and developability data provides early and actionable risk visibility. So collaboration with such as ASI ProImmune allow immunogenicity science to be operationalized within a real discovery timelines. Importantly, I think this approach did not slow the development. It enabled confident and timely progression. As a result, we successfully achieved our project milestone, and we greatly appreciate the collaboration and your handling support. Finally, I would like to highlight the business value of the early immunogenicity assessment. As you know, in vitro immunoassays require only a modest discovery stage investment, representing a small fraction of the total biological R&D cost. However, this small upfront investment enables early identification of immunogenicity risk, helping to avoid costly late-stage clinical attrition and redevelopment. Improved lead selection increase the overall probability of technical success across the AZ pipeline. Early data-driven decision also shorten the development timelines and optimize the internal resource allocation, ultimately reducing the clinical immunogenicity risk, lowers long-term safety monitoring and post-marketing burden. In summary, I think small early immunogenicity investments generate disproportionately downstream risk reduction and long-term portfolio value for our AZ, and I think for other industry companies. In summary, thank you very much for your kind support and your collaboration with our ASI. Thank you very much for your kind attention. This is my presentation. I would be happy to take any questions. Thank you.
Great, thank you. Yeah, thanks for showing how you've applied this for the project. It's very clear the steps you were taking. Do you apply these methods on all of the projects in the same way, or do you apply them differently depending on the project's needs?
Thank you for your question. So currently, under ASI, we only successfully use this integrated strategy in the current project, but we also have another pipeline. So in the very early stage. So yeah, of course we use this successful case and in another in the next project, I think.